Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. What role does quantum physics play in the brain? Quantum physics is obviously key for understanding why large molecules like proteins and DNA are stable under the heavy bombardment from the thermal motion of small molecules like the water molecule. And the formation of the strong covalent bonds that hold molecules together can only be understood by quantum physics. But does quantum physics play a more direct and exciting role in the processing of information in the brain? The most spectacular suggestion along this line was put forward by Roger Penrose in his two books, The Emperor's New Mind and Shadows of the Mind. Here it was argued that consciousness may arise from genuine quantum mechanical processes, suggested to take place in microtubules inside neurons. Francis Crick, co-discoverer of the structure of the DNA molecule, later commented with characteristic British understatement that it would be remarkable if this is true. And Penrose's ideas have not received too much traction in our community. But personally, I think it's great that people are not too shy about presenting bold ideas, as long as they are honest about what is known and what are speculations. A broader question is what role quantum physics plays in biology in general, not only in the brain. Today's guest, Johnjoe McFadden, argues that it may play a larger role than previously acknowledged. He's a professor of microbial genetics at the University of Surrey in England, and together with Jim Al-Khalili has written the book Life on the Edge, The Coming of Age of Quantum Biology. Here they explore several biological phenomena that may only be explainable by quantum physics. In the podcast, we discuss many of these biological phenomena, for example, how quantum physics can help in photosynthesis, that is the process where plants store sunlight to make food for us and animals, or smelling, how we can register odor molecules in our nose, even in very, very small concentrations. Further, we discuss whether quantum physics can help in making mutation that is required for evolution, and even help the brain making us think. McFadden is also an expert on antibiotics, that is, the precious drugs we take to fight bacteria infections. And towards the end of the podcast, we discuss how we can fight the problem of bacteria getting resistant to these drugs. Note that this podcast was recorded in November 2019 as part of my Vett og Vitenskap Popular Science podcast series. I reuse it here as a bonus episode since I believe it can be of interest for many listeners of this Theoretical Neuroscience podcast. This episode was only recorded as audio, so there are no video recordings of the conversation with Johnjoe. As for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. And if you want to support the podcast, go to patreon.com/theoreticalneurosciencepodcast and choose a membership of your liking, for example, at the cost of $1 per month. As a member, you will get access to a video version of the podcast recordings as well as episode transcripts. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast where you can watch the videos as well. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Johnjoe. So, welcome to the podcast, Johnjoe McFadden.
Thank you.
So, you're a professor of molecular genetics at the University of Surrey and also a director of a new center for quantum biology.
That's right, yes.
Well, we came across because you were coming to Oslo for a conference on antibiotics and bacteria, which is also one of your research interests. But you also wrote this book called Life on the Edge, The Coming of Age in Quantum Biology, which you wrote together with Jim Al-Khalili.
Yes, I did, yes.
Yeah. So, quantum biology, that sounds really exciting.
Yes, it is very exciting. It's to me, it's the most exciting science of the moment, and I think it could be the science of the future. And It's the first new science of the 21st century, so it's got a lot going for it.
Well, everything sounds exciting when you put quantum in front, doesn't it?
Yeah, sounds exciting, but a little bit hard as well for many people.
Yeah.
So that's the challenge.
So it's really about how to use quantum physics. I mean, the applications or the quantum physics and the quantum effects in biology, and so then it's important to first make a clear distinction. What is different between classical physics and quantum physics?
Yeah, that's kind of tricky, but it really you have to get a little bit of the history with classical physics, which was pretty much all worked out towards the end of the 19th century and the beginning of the 20th century with, say, the atomic theory and general relativity and special relativity. But then there were various things that were left out, and it was discovered that particles at a very microscopic level didn't behave like big objects. They could be in many places at once. They could be in many states at once. They could pass through barriers that they shouldn't pass through, and they could have connections despite being separated by huge distances. So these aren't really a part of classical physics, and probably most fundamentally, quantum mechanics isn't entirely deterministic in that there's a randomness at the heart of it, and this goes completely against classical physics, which is based on order.
Exactly, because in classical physics, which I guess started with your countryman, Isaac Newton, so here the idea really was that, for one thing, it's assumed that the basic building block of nature is particles, and these particles have a particular mass, and they also have a particular position. You can say where they are with as precise as you want, and they also have a particular velocity. And then, as you say, this was developed to explain, I mean, all kinds of phenomena, like gases and liquids, and also Einstein took it further to also explain this relativity when things are really heavy and we're really fast. But then, when they started to look at atoms, the smallest building blocks of nature, and they want to, because atoms consist of then this small nuclei with electrons zooming around, and when they wanted to try to understand how the electrons behaved in atoms, they saw that this classical physics, the physics of Newton, was no longer, couldn't be applied. So you got all this weird quantum phenomena, right? And I guess so you had, like, at the beginning of the 20th century, two major revolutions in physics: the relativity with Einstein, and then which has a lot to do with the big and fast, and then you had the quantum physics, which had to do with the small and light. So I guess the major in 1925 or 26 or whatever, then we had these quantum mechanical formulations of how electrons in atoms behave, and with the Schrödinger equation, and that's what everyone learns in physics. Any physics study around the world has this. Learn how to do. Then physics students learn how to do quantum physics, and then so you had this. So this was all about electrons, and then, but also then it was found out that this Schrödinger equation is not only for atoms; it's also for molecules. And biological systems are made of molecules, right?
Yeah, I think this was, to me, the most excuse me. This was the most interesting question as to how far you can take classical physics in biology. Nearly all biologists come from a training in classical science. So we learn about chemistry, atoms and orbitals and that kind of stuff. And we know vaguely that things like atomic orbitals and bonds are to do with quantum mechanics, but we don't really learn quantum mechanics because, by and large, it's thought to be that macroscopic stuff is still perfectly adequately explained by Newton's laws, and as they're applied
in animals and everything. Big
you
can see.
With thermodynamics added on, and the science of large numbers of particles, and that all makes sense. And it was thought that it goes all the way down. And then when I started reading a little bit about quantum mechanics, particularly I remember reading John Gribbin's Schrodinger's Cat, and it told me that at the level of atoms and particles and electrons, things were completely different. It was a completely different world down there. And as a biologist and a molecular biologist at that time, and still am, it occurred to me that actually life is kind of at that molecular level where it's sitting on the edge between whether it's going to be classical or quantum. In enzyme reactions, particles are moved around within from a substrate to make a product, and in respiration, electrons travel along kind of nanoscale wires. And it really is a question. Everyone was assuming this is all pretty much classical science, but there was no question to me at the time that this was 20 years ago, really, when I first started thinking about this, that this could also involve quantum mechanics.
And you wrote the book 20 years ago. That was like
yes,
the first
that's right.
book, maybe,
in fact,
quantum biology since Edwin Schrödinger's book, almost like What is Life?
Indeed, it actually linked back to what you mentioned before. I'm here in Oslo to attend a meeting about antimicrobial resistance, and this is how bacteria become resistant to antibiotics. They acquire mutations. And about 20 years ago, some kind of mutations were discovered by a scientist called John Cairns in the US, very eminent scientist who worked with Watson and Crick, which seemed to go against all the rules. They were mutations that became more frequent when they provided an advantage. And this was against all the rules of biology as we knew them then. And as I was reading about quantum mechanics, I suddenly thought, well, actually, it could be that these would make sense within the rules of quantum biology. And not knowing enough about quantum mechanics at the time, I got in touch with our physics department, and they invited me to come and give a talk about this. And I gave the talk, and it was skeptically received, shall we say, but politely. But in the audience was Jim Al-Khalili, who was at that time just working as a particle physicist, working on something called halo nuclei inside atoms. And he came up afterwards and said, well, there's problems with this, but, maybe we can work on it. And over a next year or two, we worked on this problem, and we wrote a paper about this kind of mutations called adaptive mutations, and provided a speculative quantum mechanical model of how they may be occurring. But by this time, I had read so much about quantum mechanics and thought so much about it, I wrote the book Quantum Evolution, which was making a claim that quantum mechanics was fundamental to biology. And then everything went quiet, really. Our paper wasn't picked up that much, and adaptive mutations became rather messy, shall we say, until really the turn of the millennium. And then some experiments started turning up in biology that were hard to explain by classical physics. And then maybe 10, 15 years ago, really quantum biology experiments were performed that found evidence for quantum mechanical phenomena in phenomena such as in biological systems such as enzymes, photosynthesis, even how birds fly around the globe and use a compass to know where they're going. So suddenly, rather than an admission of insanity, believing that quantum mechanics could be involved in life was considered just kind of slightly eccentric.
Redeemed a little bit.
Well, still,
it's not completely crazy.
It's not completely crazy, at least.
Exactly.
It's still in the exotic area of biology, but not completely crazy anymore.
Yeah. So because so the question is, so this is an important thing that obviously quantum physics is really key for that for understanding why molecules are formed, why two atoms come together and form these very strong bonds. So, for example, just the stability of long DNA molecules, change of atoms under all this, how they remain stable, you couldn't understand before quantum physics. We have to have these covalent bonds. So, but this is some people have called this. This is sort of like a boring, sort of like the boring application of quantum physics in biology, because now you're talking about a little bit more, sort of like more of the exotic or like very special effects of quantum physics, that how they can play a role. So what could be like the the hallmark. I mean, I think of tunneling. I think you talk about this in your book. That tunneling is something. This is clearly a quantum mechanical thing. So, what is tunneling?
Well, actually, I'll just go back a moment to. There are really three phenomena which we kind of recognize as being kind of key to quantum biology. One, as you say, is tunneling. The other is quantum coherence, or what it's sometimes called quantum superposition. And the third is entanglement. Those three are the kind of triplets of signatures of quantum biology. So the first one, then, quantum tunneling, is a phenomenon by which particles such as electrons and protons can move from one place to another without traveling through all the in-between places. And they do so because of that uncertainty that lies at the heart of quantum mechanics, that you can't know the position for certain. So that means if you know that the particles may be on the right-hand side of a barrier, an energy barrier that they shouldn't be able to penetrate, because you can't know their position, there's some probability they're actually on the other side of the barrier. And that probability isn't hypothetical in quantum mechanics, it's a reality. And if you start measuring them on the other side of the barrier, you'll find the particles there, even though by classical physics, they shouldn't be able to get there.
In some sense, if you're a quantum particle and you have to climb a mountain to get to the other side, you can just stand there and wait for a long time, and then you magically tunnel through. That's
Exactly. So that's a phenomenon studied well in physics, but it occurred to me that, say, for example, in DNA, the DNA bases are held together by things which we call hydrogen bonds. But basically, they're a single proton that's kind of distributed between two chemicals. And I started to wonder about whether they may be tunneling to other positions by this, hopping from one place to another without visiting the in-between places property of tunneling, and whether that may make a difference to how mutations take place. So that's one of the areas of tunneling. But then independently of that, a number of scientists, say Judith Klinman in the US and Nigel Scruton in the UK, started to find that protons also tunnel inside enzymes. And enzymes are the workhorses of life. They make all the biomolecules in your body and every body of every organism on the planet.
But they're a type of proteins.
They're a kind of protein, and they are catalysts. So they catalyze reactions, which means that they make them work faster. And by faster, we mean factors that can be as high as 10 to the 20, 1 with 20 noughts on the end. And that's one huge acceleration. If you could accelerate your walking speed by a factor of 10 to the 20, you could stroll across the universe in a few hours. So that's a huge acceleration. And how enzymes manage to achieve this kind of acceleration...
Because these bioenzymes are extremely efficient, right?
That's exactly what we mean.
To make sort of like artificial enzymes like catalysts, it's very difficult to compete with bioenzymes.
Yeah, absolutely. A factor of about 10 to the 6 or so is typical for an artificial catalyst. So 10 to the 20 is unknown in artificial catalysts. So how biology makes enzymes work so well has been something of a mystery, and it seems to be part of that answer, may not be all of it, is that what enzymes do is bring particles together close enough that they can make that they can tunnel from one place to another. So quantum tunneling seems to be involved certainly in enzyme reactions, both of electrons and protons, and that's part of the reason that we're alive, because enzymes manage to do this so well.
Yeah, because also one thing is that, like, of course, bioenzymes works at body temperature, like when in our case, like 35 degrees Celsius or 37. But often in like chemical processes in plants, they are much higher temperatures because they don't do tunneling. They sort of do brute force and use a lot of thermal energy to make the right molecules meet.
It's often understood by going over the particles are in a kind of well, and there's a hill between them and where they want to go. And as you say, it's rather like the mountain. If you can't do tunneling, as most people thought that biological molecules can't do, then you have to have a lot of energy to get over that hill. And that's why artificial catalysts work at very high temperatures, but biology manages to do it at very low temperatures, and at least part of that, the reason why they manage it is because they can tunnel through the mountain.
Another very, I think, interesting example you mentioned is photosynthesis, and this is, of course, the key process of all life. And like the life on Earth is due to photosynthesis because this is the way that plants sort of harvest energy from the sun, and then we survive by eating the plants or eating the animals that have eaten the plants. Right? So, can you take us through the key things of photosynthesis and key elements of photosynthesis and how quantum physics can help here?
Yeah, photosynthesis is one of the most complicated biochemical reactions or chemical reactions that we know about in the universe, never mind on Earth. So it's a complicated process in which energy, it is an extraordinary process. Essentially, light plus carbon dioxide and water can be turned into biomass. And this is what plants manage to do with extraordinary high efficiencies. And it's, as you say, it's responsible for all the biomass on our planet, pretty much. So it's an extraordinarily important and complex reaction. But the first step of this reaction is where a photon of light is captured, and it's then taken to
the photon of light is just a piece of light.
Yeah, a particle of light.
Yeah.
Particle of light is captured by chloroplasts, which are inside plants and similar structures in bacteria. And they capture a photon of light, and they have to then transport this photon of light through a kind of forest of chlorophyll molecules. So it's actually one chlorophyll molecule that captures the light, and it's then turned into something called an exciton, which is a vibrating electron. And then that has to hop from one molecule to another within this complex photosystem consisting of thousands of chlorophyll molecules. And the problem has been that if you calculate by classical means how this transport reaction should take place, where you have to transport the energy through a forest, essentially, mostly it should get lost. Mostly the energy should be re-emitted as light or heat before it reaches its destination. So mostly it should be an inefficient process, but in fact, in photosynthesis, it's got, under optimal conditions, close to 100% efficiency. And that's far more efficient than our best artificial energy transport, which normally is only its best, say, 50%. So how plants and microbes manage to perform this extraordinarily highly efficient energy transport has been something of a mystery. And then experiments early in the millennium by a number of researchers, Greg Engel at California at the time, for example, shone a laser light at bacterial photosynthetic complexes, and then looked at how that laser light was kind of reflected out of the system. And it gives you information of how the energy is being transported within the system. What he discovered is the light came out in waves, what he called quantum beats. And these waves were a key signature, and they demonstrated that energy transport, where it was thought that the energy hops from one chlorophyll molecule to another through this forest of chlorophyll molecules, that hopping isn't a hop at all. It actually travels more like a wave through the entire system. This means it's behaving quantum mechanically. A key aspect of quantum mechanics is particles have wave-like properties, and waves have particle-like properties. So in this case, this particle, the electron being transported, the electron energy being transported, was being converted to a wave to allow it to transfer by all possible routes through the system. And thereby it found its way to the reaction center. This is where it needs to go, very, very efficiently. Whereas, as I was saying, by classical means, it should mostly get lost in the forest.
Yeah, because I think is the analogy here, if say if you're like stuck on Manhattan and you have to get somewhere, you're not quite sure, you just find a museum and you don't know where it is. And then you can just start wandering around and actually you can randomly and just take left and right on any, and eventually you'll get there, even though you can sort of be a very, very long walk. So that's a classical walk. But the quantum walk then, and like in this case, then you sort of explore all paths at the same time at some point.
Yeah, exactly. So, when you're trying to go to work and you get stuck in a traffic jam, you wonder, I wonder if I had taken another route,
Yeah.
And you never know, of course, in the classical world, you can't go by two routes, but in the quantum world you can. And that seems to be what the light energy is able to do in this step in photosynthesis. It's only the first step, but it's an important step. It goes by all possible routes and thereby kind of finds the fastest way to get there.
Because it's, I mean, also with the solar cells, these new cells, which are like what's called the photovoltaic cells, it's also the same thing that you sort of light comes in and it excites this electron, and that has to be captured by sort of like what analogous to the reaction center before it falls down again and you lose the energy. So it's the same kind of thing.
Yeah, and exactly why this kind of research is important, because at the moment in artificial systems, you lose about half the energy. If you could somehow do it as well as the plants do it, then you'd have 50%, well, you double the amount of energy you can get from the input of light. So if this was possible, it would solve all our energy problems, at least for the time being. We'd have twice as much energy from light from solar energy than we currently have. So it would be a big deal if we could do that ourselves.
Yeah. So now we talked about two processes, like enzymatics. That's how to make sort of like things, the processes that of life happening at fast enough so that we actually can live and food and process energy. And then it's photosynthesis, which is about how plants sort of get light from the sun. And then it's also about sensing the world. I think this thing of olfaction, because olfaction is this fantastic sense where particularly some animals, maybe we are not too bad at it either, can almost like sense like a single molecule that ends up in their nose.
Yeah, it's an extraordinarily sensitive. Our noses are much underappreciated, but we have an extraordinarily sensitive nose that can sense very low concentrations of chemicals in the air. And other animals, dogs, for example, bears, are amongst the most have the most sensitive noses. They can smell a carcass miles away. And how we do that, both in terms of sensitivity, but also in terms of discrimination, we can discriminate between molecules that are almost identical to each other very easily, is again something of a puzzle, and a puzzle that isn't entirely answered by the current science, classical science explanation, which is a kind of a key in a lock mechanism where a olfactory molecule, a smell, something like limonene, which is the chemical that gives the smell of lemons, the characteristic citric smell, it goes into a receptor in our nose and kind of turns a lock. But there's a lot, and that seems to work pretty well for many examples of how olfaction works, but it doesn't work for many in that some molecules that are pretty much structurally identical, they have the same key shape, have very different smells, and some molecules that have very different key shapes have the same smell. So it doesn't seem to make complete sense. And there's been the idea around for a while that instead of the shape of molecules being detected by olfaction, it may be the vibrations of molecules. And that's a great idea. It's how we detect molecules in the laboratory, for example, by a technique called spectroscopy, in which you shine a light on the molecule, and the vibrations in the molecule change the frequencies of the light. You can tell what kind of frequencies are in the molecule. But how you do that in your nose, no one had any idea. But then Luca Turin, a scientist, worked out a way in which you could do it by a kind of quantum mechanical way, where the vibrations would induce the tunneling of an electron from one place to another within the olfactory molecule. And this was a speculative proposal initially, but he did some experiments with fruit flies, which are the most convincing, in which he showed that these fruit flies could distinguish between molecules like limonene that were made of ordinary hydrogen, the hydrogen we have in water, but also heavier hydrogen. And they could, or even though that heavier hydrogen, deuterium, is chemically the same as ordinary hydrogen.
It has an extra neutron.
extra neutron,
on
but
the
that shouldn't make a difference to the chemical properties.
Exactly. It has to do with just the mass.
Exactly, but the flies could smell the difference, so that was kind of at least consistent with the theory. It's still a speculative theory. It still hasn't got as much experimental evidence behind it as, say, the enzyme tunneling or the photosynthesis theory. But it's at the moment it's still a good way of explaining those experiments.
Okay, so now this is sort of now 3 processes which are then in the kind of like in the ongoing life or ongoing activity, and then it's also this thing that I think originally motivated you to maybe get into this quantum biology, this thing of evolution, and this thing of targeted evolution. So what is targeted evolution?
Well, this was, as I said, the most what inspired me to think about this, and it was some experiments performed by Keynes and others more than 2 decades ago, which indicated that under the right conditions, mutations that provided an advantage would become more frequent in the population. Now, that inspired me and Jim to get into thinking about quantum biology, that the quantum mechanics may be involved. But in fact, the phenomenon of adaptive mutations, as they were called, has become rather messy, and other things seem to be going on in this system. And although they're still out there, they're still puzzling.
So just to make this adaptive evolution, I mean, it's sort of, I think one example you have in your book is sort of if you have this collection of bacteria, and then you start starving them so they get less food. Then it turns out that of course bacteria mutate all the time, and then these mutations that make it easier to manage without food are happening more often than you would expect. So in some sense, it seems that you're just lucky. The mutations are lucky, but you're saying that it's maybe not just luck.
No, it didn't seem to be in this case. And exactly that. You starve bacteria in a way that if they have a particular mutation, they'll be able to eat a sugar that's in the medium you're starving them on. And they do mutate, and at a very high frequency. And it's not just that they increase their mutation rate generally. If you look at other genes, then they're not mutated. It's only the particular mutation that will rescue the bacteria from starving that happens at a high frequency. So that goes against all that we know about how mutations take place. They're meant to take place only randomly, and then selection, the direction, if you like, of evolution is provided by selection, natural selection, as Darwin famously put it. So it went against that principle because we couldn't. There isn't the processes that cause mutations, say radiation, ultraviolet light, chemicals, heat, even will cause mutations. They're random. They're kind of just, you can't see how they could target a particular gene. And yet in this system, the mutation that provided this advantage seemed to be occurring at about 1,000 times higher frequency than other mutations. So as I said, that was very, very puzzling. It was a big puzzle in biology at the time. What happened since then is that it was discovered that other things are going on in this system which may explain it. Without recourse to quantum mechanics. The DNA was being amplified. There were events called recombination going on where DNA was kind of swapping over from one place to another. The cells, although they were starving, they were still alive. The dead cells were being eaten by live cells. So essentially it became extremely complicated. And this is the problem with biology. Everything is complicated. So we kind of have to try to... in a complex system, you can always find explanations for even the most unlikely events. So within all of that, there may be explanations of adaptive mutations. So we kind of moved away from that idea and just asking the question whether quantum mechanics may be involved in mutation at all. And that goes back to the realization of me and Jim at the time that mutations may be caused by the motion of a proton within the DNA double helix. So now we're looking at whether that happens irrespective of whether it's a cause of adaptive mutations, whether this jumping or tunneling through the mountain may be a cause of mutation, and that's one of the things we're currently investigating at Surrey.
So, but these is it fair to say then that this photosynthesis, enzymatics, olfaction, and also this mutation, so these are sort of ways that quantum physics may actually may like speed up processes and make things makes things more efficient.
Yeah, that's exactly it. Really, essentially, what we're claiming in our book "Life on the Edge" is that life takes advantage of these of quantum mechanics to speed up. It has a quantum speed up, and this is something that's been explored, of course, in physics. Recently, for example, Google have claimed that they've made a quantum computer which demonstrates quantum speed up. That is, that the efficiency of it performing a calculation through quantum mechanics is faster than you can do it classically. So what we're claiming in biology is that 3.5 billion years ago, biology discovered this quantum speed up.
Yeah, exactly. Particularly with the photosynthesis, then maybe.
Yeah.
So, and then one fascinating example in your book is this thing of avian navigation. I mean, in Norway we have a lot of birds coming home. I guess now they have left for the winter, but we have and going for the south. But some of them are coming back. Well, all of them, we hope, in the spring. And then it's also this how do they find the way?
Yeah, this is also a very interesting story, I think, in quantum biology, and it's one that is based on one of your local inhabitants, the European robin, which I believe you.
Strupe, I think.
Yeah. It's different. In the UK, we have our robin, and it stays in the UK over the winter, and that's why it ends up on all our Christmas cards, because it's the birds you see hopping around in the snow. So the UK robin stays put for the winter, but the European robin that lives further north in Scandinavia, it travels south, and like most birds, migrating birds, it uses a compass, and actually most birds have several compasses they use. Some of them can stellar navigate, some of them use the position of the sun, they all use landscape cues, but some birds, and particularly the European robin, definitely have a magnetic compass. They use a magnetic compass to help find their way to southern Europe and North Africa, which is where the European robins spend the winter. And that's, again, quantum biology is usually focusing on those areas where it's hard to explain a phenomenon by classical science. So how do you make a compass out of a bird? Excuse me. And this, so how do you make a compass out of a bird? And if we think of our typical navigation compass, we would call them a Boy Scouts compass. In UK, it says a little magnetic needle.
Scouts of all genders here.
Exactly. Yes, Girl Scouts as well.
Yeah.
It has a magnetic needle, and it points north. But we don't have any magnetic needles in our body.
No.
We're made mostly of water. How do you turn what is mostly water into a compass? And this is a big problem, and there are many ideas going around. It's thought that birds may have some crystals of magnetite. It's been proposed in the beak of a pigeon, for example, but then that's recently been questioned and doesn't seem to be the case anymore. So how do you detect magnetic fields in flesh is the question. And a chemist called German chemist called Klaus Schulten came up with the idea first. He showed that some chemical reactions involving chemicals called free radicals, which have characteristic of free radicals is they have what's called an unpaired electron. And unpaired electrons are magnetically sensitive. So what Klaus Schulten showed is that some chemical reactions that generate free radicals are sensitive to magnetic fields in the sense of their products will be affected by a magnetic field that you apply to the reaction. And he proposed a long time ago that maybe birds might be doing this as well. But no one was able to follow it up until experiments performed again at the beginning of this millennium by Thurston Ritz, who was a student of Klaus Schulten's. He teamed up with a pair of ornithologists called the Wilschgos, and they'd already demonstrated some peculiar features of the robin compass, and that is that it requires light. It's actually in the bird's eye, and that it doesn't point north or south, this compass. It just finds the nearest pole, and that means it's probably something called an inclination compass, which measures the angle of the Earth's magnetic field to the surface of the Earth. And that angle, wherever it is, will point to the nearest pole. And this seemed to be what the bird's compass was doing. But again, no one had any idea. How do you make an inclination compass out of a bird's eye? And Thurston Ritz came up with this idea that a protein in the bird's eye called cryptochrome, which was known to be present in the retina, will, when interacting with light, generate free radicals. So he proposed that this is the bird's compass. That
What are
light.
free radicals, the molecules with the ions?
Free radicals are ions with unpaired electrons. So they have a single electron. So it's usually caused by a molecule that splits in two. And in the original molecule, there's a pair of electrons. And then when it splits in two, each of the products have a single electron. And these are magnetically sensitive then. And when they recombine in one way or another, then that recombination is sensitive to magnetic fields. So Thurston Ritz proposed that this was going on in the bird's eye. Lights are picked up by this cryptochrome protein. The protein generates free radicals, and then they recombine in a way that's sensitive to the Earth's magnetic field, so that the bird can kind of see the Earth's magnetic field because it's happening in the eye. And he then made a prediction from it, that from his idea, that the bird's compass ought to be disrupted by radio waves of the particular frequency of these free radical events. And he collaborated with the Wilschgos to test this theory by firing radio waves at the birds, and showed that they were indeed disrupted by radio waves. So it doesn't prove that that's the bird system, but it's the only explanation so far of those experiments. So, a lot of other experiments now have supported this kind of idea, and it's a very active area of quantum biology.
So what's the quantum aspect of this?
Well, oh yes, yeah. Well, the electrons that are separated, they're actually in a state of coherence or possibly also entanglement. And entanglement is one of the weirdest aspects of quantum mechanics. The one that Einstein, who remember gave us black holes and bending space-time, he said no way to entanglement.
What is entanglement?
Entanglement is where you have two particles that interact, and then you pull them as far away as you like. You could pull one to one end of the universe and the other to the other end of the universe. And in quantum mechanics, that doesn't matter. And if you poke one, the other one will jump instantaneously at the other end of the universe. And that's a big problem in general relativity. And it's why Einstein really didn't like quantum mechanics, despite being one of the people who...
Ghostly action at a distance?
Spooky action at a distance.
Exactly.
That's what I know it as. So he didn't like it, and he denied that it took place. But then experiments in the 1970s showed, that it did take place, that photons could be entangled over, first of all, distance across a laboratory, then distance from one island to another in the Canaries. And now people have done lots of entanglement experiments in lots of systems, and showed that when you separate particles, they do have these spooky actions at a distance. Still very puzzling, even in quantum mechanics. And it's a real problem with trying to fit quantum mechanics with general relativity and gravity. But so the electrons back to the bird's eye and the cryptochrome, when cryptochrome interacts with light, it generates these free radicals that are quantum entangled. And this is part of the reason why they become sensitive to a magnetic field. So entanglement, this peculiar property of quantum entanglement, may be helping birds and many other animals that have magnetic compasses, and that's a lot of migrating animals, and even plants seem to have magnetic compasses, may be using this weird property of quantum mechanics to operate their compasses.
So of these four, you mentioned photosynthesis, enzymatics, olfaction, and mutation, and also now navigation. Of those five, what do you think is sort of like the clearest sign of interesting quantum effects?
I think the ones that are soundest in terms of the bulk of experimental evidence for them are photosynthesis and enzymes, followed closely by the avian navigation system and maybe the tunneling in DNA. I think the olfaction is lacking substantial support, and it's still very controversial. But it's still out there, and we hope it's true because it's such a good story. But it needs more experiments, I think, to support it.
Good stories are not always true.
No, exactly not. Yes.
That's true. So, but I think it's like if you ask people about the biggest mysteries of the universe or in mysteries around, I think some people say, well, how did the universe come about? Why is it something rather than nothing? And another one is how can a group of atoms like we are become conscious? And a third maybe is sort of then how did life arise on Earth? I think for this, how did life arise on Earth? That happened sort of like 3.5, 3.8 billion years ago. It's not exactly clear, I guess, but at least it happened a long time ago. And everybody expects, I think, that the explanation of this, if we find it, is going to be in terms of known physics. So, but you also had some ideas about sort of how actually quantum physics may have helped, because the universe is 14 billion years old or something, and the Earth is 4.6 billion years, and then this was a very violent place to begin with, but maybe even after like 3.8 billion years ago, life was already there. So how could life originate relatively speaking, quite early? And you have some ideas that maybe quantum physics may have helped in this.
Yeah, these are only ideas. But as I said already, we tend to reserve quantum biology for explaining the unexplainable. And the origin of life is one of those events. And it's often describe it that essentially the chemistry we know about us that goes on classically is the chemistry of thermodynamics. And if you look at chemical reactions, and you can see order in chemical processes, but at the molecular level, it's all disorder. It's all atoms and molecules bumping into each other entirely randomly. And it generates some wonderful phenomena, storms and the red spot on Jupiter and these kind of structures. And it can generate structures that we see around us, like the kind of structures we'll see on ice or avalanches and all these kind of phenomena. But it doesn't really generate anything anywhere near as complex as life. And this has always been a puzzle to me as a biologist, whether simply random motion of atoms and molecules could generate the structures of life. And the way I sometimes put it is that when you take a chicken and put it into a pot of boiling water, you make chicken soup. You don't start with a pot of chicken soup and get a chicken popping out of it. And that's really down to the second law of thermodynamics, that things in the classical world just get more and more random. And you can do what you like, put heat into the system, but all you do is break things up and make things more and more random. But somehow, 3.5 billion years or so ago, an extraordinary event occurred which generated a self-replicating entity. Probably wasn't a cell initially, might have been a chemical, that gave rise to the first life forms. And that's extraordinarily fascinating and an amazing event. But how likely is it that it could have taken place by classical physics? And to answer that, you have to kind of you have to make guesses as to what's the simplest things that can self-replicate.
Because what you want to explain, right, is I mean, don't we say that there is we had like at some point we had like what's called Coluca, last universal common ancestor. That's sort of like the first sort of like self-replicating some kind of animal-like thing, and after that, Darwin's evolution can play out. But before that, before you had self-replication, so that you can get children and mutation and so on, you didn't have a substrate for Darwin's evolution. So it's really about. So is it right to phrase it as sort of at what sequence of chemical reactions did Luca arise, the last universal common ancestor?
Yeah. We can date the last universal common ancestor by looking at all living organisms and kind of writing their family tree backwards, and you get an answer around about 3.5 billion years ago to the origin of life. But then that brings us back to a simple bacterial-like organism, and you can ask, how simple can life be? And at least for the stuff we see around us today, not at all simple. The simplest life form is extraordinarily complex. It's still a bacterial cell with several hundred genes, millions of proteins, lipids. It's an extraordinarily complex organism that has more structure and organization than, say, a Boeing 747. And as Fred Hoyle famously said, when you a tornado blowing through a junkyard can't assemble a Boeing 747. No matter how hard you look and how long you wait, that's not going to happen. So what happened to generate the first self-replicator? Most biologists say, well, it couldn't have been a cell like today. It must have been something simpler, like a self-replicating chemical. The problem, and that makes some sense. You can kind of imagine a chemical that could replicate itself. The problem we have is that we don't know any chemicals that can replicate themselves in the way that life does in terms of information. We can have crystals that will form more crystals, but that's not really self-replication in the sense that life has, in which information is replicated.
Fire can make more fire, but that's
exactly. No, it's not very useful. It's not very useful. It's never going to lead to life.
No.
So, but there are these ideas that there may be a self-replicating chemical such as RNA, which is a kind of cousin of DNA, self-replicating DNA and or RNA, and even self-replicating proteins. And people speculate about these things, and they've shown that some proteins, RNA, have some properties that will be necessary for self-replication. But so far, they haven't really made anything that really does self-replicate in any environment that would be feasible on the early Earth. So it's still a puzzle. So we've been thinking a long time whether quantum mechanics may help to solve that problem. And it comes from the realization that really what is happening, what you're trying to do in terms of, if you're trying to make life, if you like, if you think about it like that, in the early Earth, it's a search problem. You've got to search through a lot of molecules to find one that can self-replicate. Searches can be helped by quantum mechanics. There's a famous algorithm called, say, Grover's algorithm, which will allow you to search very efficiently with quantum mechanics.
This quantum walk you talk about in photosynthesis is all like a search thing. You look for your reaction center.
Yeah, that's right. So we kind of came up with the idea that maybe some molecules were able to go into lots of different states. Instead of being a single kind of molecule inhabiting a single structure, it could kind of wobble quantum mechanically into lots of different structures. And one of those structures was a self-replicating molecule. So essentially, we kind of imagine it that quantum mechanics is able to go into this, what's sometimes called a multiverse of different possibilities, and that life may have emerged from this multiverse of different possibilities that exist at the quantum level, where particles can be in many places at once, inhabit many different states at once, and one of those states may have been a replicating state. So it's a very speculative idea. It's not even really a theory because we don't really even know how to test it at the moment. But it's
so the idea is that, I mean, as you said, the quantum Schrödinger equation and the quantum wave function sort of explain. I mean, that accounts for predicts a lot of different possibilities, and you're saying that you're speculating that somehow if they ended up with this possibility that was self-replicating, then somehow the system would sort of like disentangle or collapse or something, and then this is the winner, and then everything else sort of It's sort of
Yeah,
like in an observation in a,
yeah,
like old quantum
yeah,
physicists would
and
say.
that's the kind of way that a quantum computer is meant to work. Quantum computers will look at all possible solutions, and then the trick is to find the one that's the right solution. And what we're kind of saying is the universe might have been like a quantum computer operating at that time to find the right solution to the problem of making a self-replicator. So it's a speculative idea, but life is a tricky problem, and there's no one who's got the answer
No,
to it. So
exactly, and
it's a
problem. Some of these
possibility.
questions, the answer are going to be complicating and baffling. It's just
Yeah,
the question of what baffling answer will it be?
exactly.
Yeah.
Yes.
So I think
Yeah.
that there's no simple solution. So also when it comes to they also have one chapter about the mind, and I think here Roger Penrose, an extraordinary good British physicist, also had this sort of like this book, Emperor's New Mind, I think, and more some books along those lines, where he argued that the mind had, in order to understand the mind or the brain, you had to invoke quantum physics and quantum coherence and some other, at least like the kind of thinking that our brain has, and maybe also consciousness, that or at least the kind of thinking we have sort of have to do with this problem of measurement physics in quantum, the measurement problem in quantum physics, when sort of like the wave function collapses. And this theory is sort of I saw that when Francis Crick, one of the co-discoverer of the DNA molecule, talked about this theory, he said it would be remarkable if this is true. And I think also you would say that this is sort of not the kind of quantum effect in the brain that you find most likely.
No. In fact, when I wrote Quantum Evolution all those years ago, back in 2001, I had planned to a chapter exploring this theatre, exploring the Penrose Hammerhoff. Stuart Hammerhoff is a very eminent anaesthetologist in the US, who is partnered with Penrose to champion this theory. And the more I looked into it, the more it didn't seem feasible to me. I mean, the kind of systems that I've been talking about that may exhibit quantum mechanical phenomena in biology, they're very small. Say, within a photosystem where an electron may be moving in different places by many different routes, or tunneling where a proton may tunnel from one place to another inside a single enzyme, or in DNA where a proton moves from one side of a double helix to another. But it's always on a very, very small scale. To get the kind of thing that Penrose and Hammerhoff are talking about, you have to have quantum coherence, as it's called, where a system behaves quantum coherently, at the scale of the brain. It's got to happen over large areas of the brain, because what they're trying to do is solve really what is a tricky problem in understanding consciousness. How it is that if you looking outside to the fjords of Oslo, and all of that information that I'm seeing in one instantaneous moment of consciousness is encoded in different parts of your brain. There's neurons firing in one place that will be encoding some information about a tree, another one a cloud, and another one somewhere else, the sun. And all of this is distributed amongst different neurons in different parts of your brain. And the big problem you have in understanding consciousness is that that's not how your conscious mind is. It's all kind of joined up in an instantaneous moment of consciousness. And what Penrose and Hammerhoff claimed, well, quantum mechanics can do that. And they're right, quantum mechanics can. You can bring together all this information in a quantum mechanical system, and it gets all kind of stuck together as a quantum coherence or entanglement or superposition. But it's really, really, really hard to think how that could work across the brain, the kind of quantum mechanical system that they described. And it's really Hammerhoff who came up with the idea that there are structures. Well, we know there are structures inside neurons called microtubules, which are long strings of protein, and they may connect across the brain, maybe sometimes going from one neuron to another, and that somehow they may form what's called something like a Bose-Einstein condensate, where which you normally have, which if you want to make in matter, it's a quantum mechanical state involving lots of atoms, but you have to cool something down to close to absolute zero and in a
Minus
vacuum.
270 degrees. And
Exactly.
it's just like very.
And it's only a handful of atoms or something that you get to go into a boson condensate. To get that in the brain, after I'd been reading enough about quantum mechanics, and I knew how hard quantum physicists have to work to generate systems in which demonstrate quantum biology at a larger scale, quantum physics at a larger scale, how difficult it is. And the idea that this was operating in a hot, wet brain just seemed infeasible to me. So I thought that this was it just doesn't work quantum mechanically. And I don't know any quantum physicist who believes it. It's just not possible by anything that we know about quantum physics. So we don't. I don't believe that theory at all. No.
But you still, I think you mentioned that there has been some, even though this is sort of my research field, you also point me to some work which shows that maybe some of this, like this, the action of sort of like these ion channels, the currents that goes through cell membranes can be also get some kind of enhancement by quantum physics, but that will not sort of address consciousness. It will be more about efficiency again.
Yes, I guess so. You could have ions traveling in ion channels that may travel quantum mechanically through the ion channels, but that just makes the whole process a bit more efficient. It explains why it's more specific as well, why ion channels are specific for particular ions, but it can't on its own explain consciousness, no.
But in some, because you're very interested in consciousness, and who isn't? Everybody who's
everyone who's conscious has an interest in it.
This is a mystery, something which is there, and we very much would like to understand. And you have some ideas about this, that is electromagnetic. I mean, the field in the brain, because just like our brain consists of like roughly 100 billion nerve cells and some other support cells, and they occupy about like maybe 80-90% of the volume in the brain. But between there are like liquids, and these liquids are what's called electrolytes. They can conduct electric currents, and there will also be electric fields there. And this is what you call sort of like referred to as the electromagnetic field. We often call it the extracellular field, because that sort of is outside cells. And actually, our group here in Norway has done quite a bit of work on modeling this extracellular field. So that's how actually you contacted me before coming to Oslo to discuss these things. So what is your ideas there?
Yeah, well, it goes back to I came up with this idea when I had I was writing this book about quantum biology, quantum evolution back in 2000 or so, and I had planned a chapter on consciousness going through the Penrose-Hammerhoff idea. And the more I read about it, the more I became skeptical. And I thought, I can't really make a convincing chapter about this because I don't really believe it. But what Penrose did convince me in reading his book and other people's work is that there is a problem here of how you integrate information in the brain. As you said, the information that I'm looking at, that I described a moment ago, it's encoded it within a significant fraction of those 100 billion or so neurons. And those neurons are distributed throughout the brain, and yet we perceive it as a single moment of our perception, a single perception. How do you bring together all that information? Now, Penrose and Hammerhoff have proposed that it goes together through matter forming this Bose-Einstein-like condensate. But it just occurred to me that actually every time a neuron fires, it generates an electromagnetic signal. It generates if you like, a wave. And that is a wave of information that carries exactly the same information as the neuron firing. But what happens with that wave is it combines with all the other waves to generate, as you say, the electromagnetic field of the brain. It's the field that you detect by EEG. Remember those electrodes that people put on your skull to see what's going on inside your brain? That's the electromagnetic field in the brain. And it unifies all of the information in the brain, just like if you drop lots of stones into the sea, you'll see the waves all forming together and forming a kind of unified wave. So waves in the brain will unify all the information in the brain. And it just occurred to me that if we just go from the matter of the brain to the electromagnetic field of the brain, and we say instead of locating consciousness in its matter, the brain's matter, we just say, well, why isn't it in the brain's electromagnetic fields. They're still physical. They're as physical as matter. Einstein's famous equation, E equals mc squared. You have energy on one side and matter on the other. So they're just as physical as matter, but information within electromagnetic fields is unified in a way that matter isn't. And if we switch from going from the matter of the brain, which obviously is responsible for much of what the neural information processing that goes on in our brain is taking place, all of that, most of that is going through all of those neurons, through all the matter, doing all the kind of things that computers do very well as well without consciousness. But alongside that, there's the conscious electromagnetic field, as I call it, where all the information is brought together. And this, to me, is the simplest solution to consciousness. Just move it into the electromagnetic field, and most of the problems of consciousness disappear.
Yeah. But this is here's quantum theory is not necessarily a part.
No, it doesn't necessarily require quantum mechanics, but I think, fields, you can describe fields by quantum mechanics, quantum field theory, of course. So there is both a classical and a quantum mechanical description of fields. It's not that there's phenomena, unlike with matter, where matter may behave entirely classically at a macroscopic level. Fields always have a quantum and a classical aspect to them as well. You can describe fields by Maxwell's equations, which are classical, or you can describe them by quantum electrodynamics. So there's quantum mechanics there, and I think it's an interesting question which provides the best explanation of consciousness, whether we have to move to the quantum mechanical equations or are the classical sufficient. And I'm not enough of a physicist to be able to answer that question.
So I just want to. I mean, these are of course speculations, but what I really like about this book, because it's. I mean, when you hear about quantum biology, you can maybe get a bit suspicion that this is just a bunch of hogwash. But this book I can highly recommend because it really takes you to the background of quantum physics, and it's really clear about what is known and what is speculations. And I think it's very important to start thinking about these things. But as you said, that's quantum biology. These questions has been a little bit taboo to discuss in science. As sort of not that it's been, but it's not. It's been difficult to get funding. Somehow it's not been considered important. And also this is a little bit sort of flaky on the lunatic side. So I think it's. But this is certainly something that should be explored. And I'm very happy that people like you and are taking it up. So I would very much recommend this book. But before I let you go, I also you are here to talk about antibiotics. I mean, in a conference and discussing antibiotics, and this thing about antibiotic resistance is something that worries everyone. Also us in Norway, of course. And so the thing is that if you use a lot of antibiotics, then the bacteria around sort of adapt, mutate to deal with these antibiotics, and then after a while, these antibiotics are no longer efficient, and you start getting these strains of disease, types of, for example, tuberculosis, who are no longer where antibiotics no longer efficient.
Yeah, it's a huge problem across the world where something like 500,000 strains of drug-resistant TB are turning up, and what that does is you have to. If a patient comes in with drug-resistant TB in, say, Norway, then we have the facilities to be able to treat them. It becomes more expensive. Patients have to be often
what is the
isolated.
Then, if they're not, they just have to
Well, it's additional drugs. Already TB is difficult to treat. You have to treat with 3 or 4 drugs for 6 months or a year often. And for drug-resistant TB, you have to add on more drugs. And those drugs tend to be more toxic, so you have to monitor the patients more closely. These patients are infected with more dangerous strains of TB, so you often keep them in isolation as well. So it's much more difficult, it's much more expensive, it's much more difficult for the patient to live with. And that's in Oslo or in London or in New York. In the developing world, treating someone with multi-drug TB, it's often a death sentence. So particularly in the developing world, it's turning a disease that is treatable into, in many parts of the world, an untreatable disease. So it's kind of, we're essentially eliminating the advances that antibiotics have provided us, which is a huge advance in medicine in terms of people don't die of these terrible diseases that we used to die of in huge numbers from infections. And by and large, in the West, most of those infections can be cured with antibiotics, but we're losing that advantage.
Bacterial infections, which is like
Exactly. All bacterial infections are now prone to antibiotic resistance. Methicillin-resistant Staph aureus, for instance, MRSAs are a big problem in hospitals. And in TB, as I've mentioned, in gonorrhea, it's a big problem. So there are many, many infectious diseases where it's becoming more and more difficult to treat. And if that goes on, we will, and we're not developing enough new antibiotics. For example, in the TB field, the antibiotics, the 4 antibiotics we use at the moment for standard treatment, they were discovered 50 or more years ago. There's a couple of more of new antibiotics that have been discovered in the last decade ago, but we're losing already resistances coming in those, and we're losing our ability to keep up with antibiotic resistance.
Why is that? Is it because there's not enough research? Or?
Yeah, essentially it's becoming harder. We've discovered the easy antibiotics, if you like, and it's become harder to find the more antibiotics. It's very expensive to discover antibiotics and to test them and to develop them into drugs. It costs a lot of money, in millions of pounds, often hundreds of millions of pounds to develop an antibiotic. And we've just fallen behind because we've trusted the antibiotics we have to keep things under control. But that is increasingly no longer a good bet, and we have to invest more into discovery of antibiotics, but also work out ways to deal with a lot of infections without so many antibiotics. We use antibiotics in a lot of different places that we don't need to necessarily. For instance, in livestock production, where antibiotics are used on the farm, we have to try to cut back on that. Have to try to cut back on giving antibiotics to everyone who has a fever, which happens in many places, that you give antibiotics just as well. We might as well, and have to try to keep our use of antibiotics as to restrict it to only those situations where they are really going to make a difference.
Because that's something. Because I mean, if we start, say, using less antibiotics in Norway, it's also that then the bacteria adapt again, and so that they, in some sense, then these antibiotics then become efficient at a later stage.
Yeah, it's been shown that antibiotics, which we've kind of abandoned 20, 30, 40 years ago because of resistance, now bacteria are becoming sensitive to them again. So you lose the resistance if you stop using the antibiotics.
So it actually makes sense to do this. It actually helps. It's not just something.
Absolutely, yes. Yeah, it's very important. And so it's a really big problem. It's one of the major problems for medicine, particularly globally, where in some parts of, say, Eastern Europe, instead of, in Oslo, maybe about 1%, 1 or 2% of TB strains will be resistant to antibiotics. In some parts of Eastern Europe, it'll be 50% of strains.
So it's like different bacteria in different places.
Absolutely. In places where the treatment has not been monitored so well. Why you give 3 or 4 antibiotics is because if you do that and you treat for 6 months, you kill all the bacteria. If you can only afford, say, in a clinic in a poor country, maybe 2 antibiotics, well, you'll take 1 or 2 and you'll hope for the best. But mostly the best doesn't happen. And what happens is, instead of curing the patient, you make the strain resistant to the drugs. So if you don't treat properly, which requires care, it requires expense, it requires the patient being properly monitored, then you develop antibiotic resistance, and in places where
make matter worse, both for the patient and the society.
Yeah, absolutely. It's most antibiotic resistance is caused by poor medication. Medication that isn't completed, medication that isn't monitored properly, unnecessary medication. So it's a problem that we have to try to control, and often it means putting resources from wealthy countries into poorer countries, because those people from poorer countries are going to be coming to wealthy countries to work, and we have to deal with that problem by helping those countries to treat patients properly, so as we're not developing drug resistance around the world. Because bugs don't have borders.
No,
They just pass through borders. They're invisible. Borders are invisible to bugs. So we have to realize that, and realize that if we want to keep our drugs for ourselves to treat the kind of infections that we suffer, we're going to have to deal with that not only in our own countries, but in the countries where resistance is becoming more and more common.
And then it's also important when we get then like antibiotic treatment that we actually take the whole treatment package, so that you really make sure that you kill the bacteria. If not, you might risk ending up with you kill almost all, and the one you don't kill are the one who are.
That's exactly it. Essentially, if you don't take the antibiotics for the right length of time, then all you're doing is selecting for resistant strains.
Yeah.
So you're performing evolution in your body of evolving bacteria towards resistance. And if you take the full dose, then even those, then you'll stop that happening. You'll cure the disease before you get any resistant strains. But if you don't take them all, then that tiny fraction will be enriched for resistant strains. So you'll become a little evolutionary laboratory to develop resistance.
So this is follow the doctor's orders.
Exactly.
Yeah. So actually, we're talking about bugs. I just heard this other this story that actually ants actually can help us maybe in developing some of these new antibiotics, because supposedly, I mean, in this hive, in this ant hive, they have all this like problem with. I mean, ants have problem with mushrooms, and there's all these things being brought in from the outside to the hive. So in order for the ant hive to deal with that, they've developed all kinds of defense mechanisms to deal with all kinds of like, so that there's sort of like, at least that's, people are arguing that it's one of the reasons why we need to make sure that you have don't kill off all the insects, is that they actually also is like this like source of possible antimicrobial.
Absolutely. There's no, I mentioned that it's harder to find new antibiotics. Most of the antibiotics come from soil bacteria. They're bacteria that are growing in the soil. And we've harvested most of those that are easy. But people are going to all sorts of different places, like you say, ant colonies, down to the bottom of the sea, edges of volcanoes, to find bacteria which are harder to grow and may have antibiotics that may be useful for us in killing these antibiotic-resistant strains. So nature is the reservoir of antibiotics.
Exactly.
So preserving nature, we're preserving our defense against the microbes.
Exactly. So biological diversity, and why like the rainforest, for example.
Exactly, yeah.
So treasures there for...
Soils and ant colonies, etc. They're the rainforests that have all of the antibiotics that we may need in the future.
Excellent. Well, thanks a lot for coming to the podcast.
It's a pleasure.